Okay.
Whenever you're ready.
Okay. So if checking the attendance just now was a test for me to try to remember all of your names in 2 minutes, I just failed that test. Usually my students at the teaching evaluation, I often get comments that she remembers all our names very, very fast. But certainly I can't do that in 2 minutes.
Yeah, I'm definitely not gonna get that comment.
But still, I'm very happy to be here today. I envy all of you. I didn't have such an opportunity when I was an undergrad, but here I am. I heard you guys are interested in doing some undergrad research, and you want to look into undergrad research opportunities and see what this might offer you in terms of career development. So I'm happy to answer questions. First, let me just give you a very brief intro about myself. So currently I'm an assistant professor in the Department of Physics. My research area is biophysics. Essentially, we use highly advanced microscope to look at the inner structures of cells. So the cells are, in our body, the cells are on the order of, say, 20 microns, and they're finer details. You may have learned it in your high school biology. There are these different organelles, mitochondria, nucleus, and recently people found that there are proteins that can self-assemble into micrometer scale structures. Usually you would think of proteins inside the cells as being diffuse. So they should be everywhere, more or less uniformly distributed inside the cells. But people found recently that there are these proteins that can spontaneously just assemble into micrometer scale clusters, and they have important functions for normal physiology. And when the assembly is misregulated, that can cause severe diseases like neurodegenerative diseases and cancers. So my lab uses these microscopes to look deep inside the cell to see how these proteins assemble. That's what we do now. And before I came here, I think my academic path is an interesting one. Right now we do very interdisciplinary research, and that's related to my background. My undergrad training was in chemistry and physics, and then in my PhD, I studied materials. So I got a material science PhD. I was again using imaging techniques to look inside the biomaterials or soft materials to see how Brownian motion happens in those. So it's quite fundamental from a materials or physics perspective, but it has not so much to do with biology. And after my PhD training, I went to University of California, San Francisco, because towards the end of my PhD, I went to a conference. I met an interesting professor there, whom I thought I could work for and really apply my imaging techniques to biology. So I went to his lab and started to use a microscope to look at the inner life of cells. And then I came here about 2 years ago, and now I have a lab. I have 5 people working with me. 4 of them are grad students and 1 is a postdoc. I think everyone is happy in our lab. Of course, they may say different things, but that's my perspective. I'll just kind of just pause it there and let you just feel free to ask any questions about anything I said or anything you feel that I may be useful in helping answering.
Go ahead.
So was it when you went to California that you discovered that you could use physics to explore biology or vice versa, or did you know that beforehand?
I think that's a great question. I think I had this idea. I know it can be useful, but I didn't quite know in what way it can be useful. And in research, it's also a very competitive career. You do have to find your unique strengths, because broadly speaking, everything is connected, but you do have to identify your niche. So I thought that was, in the end, it was a good decision. My postdoctoral lab was very nurturing environment. I learned a lot about biology there, and I was able to apply my my physics and chemistry expertise to study these questions in cell biology. So that was a good combination. Yeah. So you kind of have to, I guess, in a way, fumble your way through, even, after you get a PhD, there's still a lot of things that you can learn and can explore. Yeah.
Hubert.
So my name is Hubert, and I am also in the physics field. So as you were saying, I understand that it's very competitive in the research field, and you're saying you have to find your niche quickly. So I feel like there's a pressure to find focus very early on and everything like that. So I understand it probably must have been very hard, difficult to switch your focus from material sciences to swimming and biophysics. Could you tell us a little bit more about how you made that decision and how difficult it was for you to transition?
Yeah, that's a great question. I would say there are so many interesting things out there. You just can't study everything, so you have to start somewhere. Sometimes it's just about following your intuition. You find some labs research that you don't quite understand, but you feel it may be interesting and maybe something that I can do. So you reach out to the lab, and then you may start working there, and you realize it either fits or doesn't fit. I think as an undergrad, you're allowed to explore these opportunities, and you have time, right? You can work in the lab for a few months and then decide if you want to continue or you want to switch to something else. There's plenty of time to try out. And I think, in a way, doing research is about understanding yourself better, knowing your strengths and your limit, and see what are the things that you can work on to improve, what are the things that you're already good at and can build on to establish your competitiveness. For myself, making the transition from material science to biology is certainly, there was this deep learning curve. And at the beginning, I had to work very hard in the lab. But I know it's something that can be done. In a way, it's not like I studied biology first and then tried to learn relativity. I think that would also be a steep learning curve, and in a way, maybe harder. You just have to have the motivation and feel that the effort can be paid off in the end. Yeah, I think that's true with learning anything, right? At the beginning of a semester, you learn a course. It's always hard at the beginning, but usually you get over it. You learn something in the end.
Jump to one of the pre-written questions while people are thinking of the next one. Sarah Peralt asks a more general question. What would you say is the most important skill for a researcher to possess?
Important skill. I don't know, this question may have been answered many times in this class. And I'm a bit taken aback by this question because my personal perspective is the technical skill is never the most important thing. I think perhaps your own interest and knowing yourself is the most important thing. And then the technical skills and anything like that comes afterwards. But in terms of technical skills, so for people in my lab, they work in an experimental lab setting, but other times you may be working on more computational things. I would say paying attention to detail is an important thing because you have to have the eye to appreciate the details that you may notice something else that other people have not thought about before, and that's the opportunity for you to be deeper. So you have to be attentive to details and kind of just pay attention to these things. And I don't know, like being persistent, I guess it's not a skill, it's more about attitude.
Yeah.
Yeah.
It's a good practice, though. Zach?
So I guess thinking about the question, I don't know if you accept undergraduates, but if you did, do you accept that?
We actually have quite a few undergrads
Okay.
working with us right now. And
Ah.
some of them have won prestigious awards. Dr. Wilson knows one of the students who recently won the Bristol Myers Squibb Scholarship.
Wow. But what was I gonna say?
You were just taken aback by the scholarships.
When you consider undergraduates that want to work with you, do you more focus on, because you mentioned that technical skill is a lot less important, or maybe less important, at least to you. Would you consider a candidate who is less technically skilled, I guess, or like less experienced, but has more motivation and is more interested in your project?
I think as an undergrad, for like at our like technical level, the way we think of undergrads are like a piece of blank paper. We some are, yes, some are slightly more experienced than others because they have, I don't know, done some research in their high school or have worked in a lab before. But at least for me, I don't consider those as critical or even necessary. Instead, I think your interest and your academic background does matter. You know, if you're struggling with courses, then we don't want you because we know that research will take away some of your time. So if you cannot manage your courses well, then we feel that you should perhaps spend more time studying because GPA at the end of the day is very important for a lot of things, right? For grad school, for med school, for jobs. But if we think you are doing reasonably well and you have the motivation and interest, then we would consider you. Yeah.
Yes.
My name is Alexa. So I saw that you've done some work with CRISPR in your lab. Could you just tell me a little bit of like how you got involved with that tool, what you think like some of the really cool things are about it, and maybe some future applications that you might use it for in your lab?
Okay, can I throw a question back at you first? So you mentioned CRISPR. Why is that something interesting to you? And you may have already learned something about it. Can you tell me what you know?
Yeah, I'm actually reading the book Codebreaker, which is about like the development and funding of the CRISPR tool. I'm just really interested in gene editing and how that can be applied to a bunch of different fields.
So can you tell me what is Gene editing?
Yeah, it's, I mean, from my basic understanding, it's the process of going into the genome and altering a certain gene in order to produce a more desirable outcome and expression of that gene.
Okay, very good. So I don't know for the rest of the class here, I would just still say, so yes, gene editing is essentially you're editing the gene. And you can see that gene editing has enormous therapeutic potential and the research potential because you can change the genetic code. And again, for our lab, we try to find a niche in this very hot research field. I think a majority of the CRISPR effort, this gene editing effort, is on the therapeutics. They're like, I don't know, multi-million-dollar patent filed on the technique. There are several emerging biotech companies that work on these techniques, and some of them are associated with these prominent inventors. And I believe a few years ago that gene editing, especially the CRISPR techniques, received the Nobel Prize in chemistry just a couple of years ago. So for us, we're an imaging lab, and we try to do something relevant for imaging. So I identified this problem for imaging proteins inside the cells. Most of the time, so majority of the cell biology research using imaging protein would involve using plasmid. Some of you, if you have worked in a lab and have done some molecular cloning, you may know that typically people use the plasmid, things that you grow in bacteria, and the plasmid encode a protein, and then you try to deliver the plasmid to the cell, and then the cell can express the protein based on this plasmid you give it, right? So you give it the genetic code, you give it the blueprint, and then it makes protein for you. And that's, I would say, 99.9% of the research out there imaging protein is done. But the problem with that method is really this is an artificial way of introducing protein. In the most extreme scenario, the cell doesn't necessarily express this protein at all. It has no, like, in its native cellular regulatory network, it doesn't have to express this protein, or it doesn't express or express it at a very low level. But when you introduce the plasmid, you're just giving this artificial input. It could be totally taken away from the native context of the genome. So we try to use CRISPR to image protein in a better way. So the way we do it is, again, because we can edit the genetic code, we can directly go into the genome and find that protein of interest, and then we insert a fluorescence tag at the genomic site. The advantage here is then whenever this cell expresses this protein from its native promoter, from its native gene regulatory network, then this protein is tagged. So it's a much better way to understand the native environment of the protein. So that, again, it's something we try to identify a niche. We want to do better imaging, and we want to use the gene editing technique. So we came up with this, and then the lab is using that to investigate a lot of important biological processes using the native protein, which some of the questions we're asking, you just cannot answer with the traditional plasmid or other transgene way. Did that answer your question?
Thank you so much.
Yeah.
Yeah. Celine?
So I just saw that you got this huge grant from the National Institute of Medical Sciences. Congratulations, first of all.
Thank you.
But what's the process of maybe going through like the grant, like obtaining a grant or finding out about grants?
Mhm. That's a great question. I think, again, in this competitive world, of course, as a principal investigator, I need to think about grants. But I think as an undergrad, if you can identify some grant opportunity that will also be helpful for, to improve your CV. So I'll just say a few things that I think might be relevant to undergrad getting grants first. So there are these fellowships opportunities out there. Those usually comes in a Dr. Watson knows better than I do on this. They comes in a form of fellowship, maybe 2 grants a year, to support you to work in someone's lab. But I think the monetary value is there, but I think it's more about this being an honor, and it's the line that you can put on your CV. I don't know what's the best way to identify these opportunities. And for my student who got that fellowship, I think we just got it from some email list. Yeah. And in fact, in our case, we were debating whether to apply, and I wrote to you and you said we should try, we could potentially be a good fit.
Yeah.
Yeah, and I know there are also those travel award opportunities campus wide. Yeah. So those are I don't know what's the best way to identify those, but I'm sure those opportunities are out there. And because a lot of your peers are not paying attention to these opportunities, so if you can take advantage of that, it would be great.
Thank you so much.
Yeah, that's absolutely true. And yeah, so you can always contact me, because for Beyond 120, that's what I do, is find these opportunities for you, or the Center for Undergraduate Research would be the other point of contact for undergraduate awards. We have one to do this November 6th, small travel award. If any of you want to go to a conference, apply for that one. As Dr. Guan was saying, sometimes students don't apply for these and then they go unused, so.
Yeah, and I would say, in a way, this is just like a lottery. You just have to keep trying, and you may get one, and if you get one, it's great.
Yeah, if you bring it again.
So, one of the things that I found on your web page was that, one of the reasons, that you switched into biophysics is because you realized you use your quantitative modeling and imaging, skills to, interested in biophysics problems. What are some of the novel research or novel biophysics concepts or problems that you encountered during your transition that inspired you to go into biophysics?
What are some of the questions can you repeat the last bit of it?
What are some concepts or novel research that you found interesting that inspired you that this field could be something that you could go into?
It's such a great question. I think initially when I thought about going into biology, applying the quantitative strengths of the physics and chemistry, I think I was mostly reading interesting papers, and I found these papers, they do research at the interface between the traditional biology and other more quantitative and engineering discipline. And I thought those papers were interesting, and they provide new insight into the biological system. I don't recall any specific system that just stood out. Perhaps that's a collection of 10 to 100 papers that I saw. I just felt there is enormous potential in trying to bridge these different fields. Yeah. And in our own labs right now, we do use these quantitative approaches to study biological questions, and we have interesting discoveries in the lab, not on a daily basis, but still pretty frequent, and we have some interesting findings.
Yeah,
I'm Megan, and so my question is, you said when you were throughout your years of the PhD, you worked mostly with physics and maybe chemistry and maybe like material sciences. But when you got involved in like the biology aspect of biophysics, how did you go about learning like all the like terminology and like the sciences? Did you like have to teach yourself, or did you find like a mentor or so on?
Yeah, I think one important ability people acquire during grad school is I think their learning ability is greatly enhanced. Nodding from another PhD in the room. You do have to learn a lot of things by yourself, and you have to learn them quickly. I personally felt that my learning ability has improved a lot once I entered grad school compared to how I learned things in as an undergrad. And for me, I remember even when I started my postdoc, like during that transition, I was finishing up my PhD, and I knew I already contacted this professor that I wanted to work for as a postdoc, and I told him, are there things that I could like reading materials that I could prepare myself? So he recommended a classic textbook at that time, which these days I recommend it to my grad students. And that book has maybe 1,000 pages. It's a classic textbook in cell biology. And then my postdoc mentor at that time said, when he started postdoc, in fact, I think he also has an interesting like transition from being more engineering to more biology, and he also made that switch during his postdoc, which is why I guess I resonated with him a lot, and I chose to join his lab. And he told me he was reading that book when he started his postdoc, and he said he read one chapter per night, so he finished that book in like a month's time. And so I think in a way, when you switch field, you try to pick up new knowledge from a different discipline. Sometimes it's very helpful to really get down to the basics and just learn it in a systematic way, maybe through reading a textbook. And you have to do that in a persistent way. Everyone is busy, and being a postdoc, you have a lot of things to do every day, but you still have to just be persistent and allocate, say, 2 or 3 hours a day to try to read into a textbook to learn the skills. So you do have to, I think if you really want to learn something new, then part of it is about planning and time management. You have to allocate the time to persistently learn something. Does that answer your
Yeah.
question?
So first of all, just like congratulations on the grant. I saw that on your site.
Thank you.
It's very impressive. But one thing I was noticing when I was looking through like all the papers you published is that you tend to publish about like 1 a year. Is that just like how the field is in biophysics? Is it like projects you get involved in? How's that kind of rate?
This, I think it also depends on the lab style, whether you like to publish you want to do a thorough work and publish a longer, more complete and perhaps more impactful story, or you like to take in small steps to publish. So I would, I personally don't worry so much about the numbers of papers we put out every year. And it's also some years, you know, as when I was a trainee, some years we don't publish. Yeah. Because if you started doing undergrad research, you'll know, you'll be very lucky if you can publish one paper, even if you start as like a freshman or a sophomore. It takes time to publish, and it takes time to write out the paper and to send out for reviews, and there's lots of uncertainty in that. So I would say maybe especially for undergrad, try to enjoy the process, instead of focusing too much on whether you can get a paper or proof things out. And I think this, yes, if you get a paper, I think it helps with like grad school application and med school application, but it's not critical or required as an undergrad to apply to grad school. I think the research experience you had and the skills you learned, and more importantly, the things about research in general that you understand can all go into that essay called statement of purpose, and I think that's an important part of the application.
Thank you. To go to one from the list, Sarah Garfield asks, have you ever felt discouraged in the research process? And maybe if there've been setbacks or challenges in the lab that you could speak to, and how does one deal with those?
I think in doing research, the setbacks are very frequent, maybe on a day-to-day basis, because I think most of the experiments fail, and that only a small percentage of them work, and then, you know, a lot of the time it's about trying out an optimizing condition, and before you get to the optimal condition, the things always fail. So it can be a bit frustrating. And how do you get over that? Sometimes you don't. That may be shocking to you, but sometimes you just have to realize that this experiment, given our current ability, our meaning, you know, the humankind or the scientific community, it's just formidable that we have no way to overcome it. Maybe, you know, when something else happens, then that part can be borrowed over and solve the problem here. But right now, there's just no way out. So you have to decide, we have to move on from there. And so what I learned from my postdoctoral training and what I tell my current student is, when you design an experiment, you want to design it in a way that it will fail fast. In a way, it sounds very counterintuitive. But if something in the end will fail, then I think I'd rather know that sooner so we can move on, right? So, because we have limited time and energy, we want to concentrate that to the most important thing and then the most practical thing, right? So if it has to fail, then let's find out as quickly as possible and then move on to something else. So sometimes you don't get over it, you just leave that and then move on to something else that are more exciting. Other times you just before that, you try very hard, you may do a lot of literature search to see if there are already published ways out there or similar ways that perhaps can be borrowed to resolve this. Or like UF is a big campus, lots of experts on different areas. You can try to see if they're local or global expert that may be helpful on this question that can help you. So you try to find all the resources you can to see if you can solve it. If you cannot, still after a lot of effort, and you just have to call it a day and move on to something else.
Yeah, Valerie.
So you mentioned writing your statement of purpose for grad school admissions. I'm wondering, how did you go about that process, like deciding what you were going to write about and writing it?
So can I ask what year you're in?
I'm in my third year.
Okay, so I think next year around this time would be like, if you're thinking of grad school, that's when you prepare these essays. So how do you go about, are you talking about the whole application process or just about the
Just
statement of purpose?
the statement.
I think that's something you just have to be true to yourself. You have to, it has to be a statement that comes from you and that touch you first before it can move the other readers. You have to identify, right, the purpose. So you have to identify your passion, why you think going to grad school is the right thing for your overall career. So I think a good strategy would be to place the grad school not as an end point, as the final goal, but as a intermediate step towards something else. This way, I think it not only helps you to overcome the tunnel vision, to have a bigger picture about where this will fit into your overall career goal. But also, I think would be more convincing if you can show that you have thought more or thought further, deeper than some of the other folks writing the essay.
Hubert again.
Third time charm.
Well, I was wondering, how did you decide to apply to grad school, with the research and the career path?
Actually, I did my in fact, I started my grad school in chemistry at MIT. I ended up not finishing that. I stayed 2 years there, and I think I did okay research. I was productive. I was nearing publishing a paper. I had a thing written. I also had a master's thesis written, but I just didn't find it very interesting. And then I found another professor at University of Illinois, whose research I really liked at that time. So that was the material research. So I reached out to him, and he had openings, so I agreed to join his lab. And at that point, it was, in a way, it's about which department was easiest for him to place me in. So I ended up in material science. But I think sharing this part of my experience is to assure you that even, when you're in grad school, you're still allowed to try out things and find the best fit and match for you. It's like you go to a department store and try on clothes, and in the fitting room, if it doesn't fit, you return it. Yeah.
So another submitted question. You mentioned having PhD students and a postdoc and some undergraduate labor. So how do you go about managing a team of people, and do you ever have to mediate conflicts?
So far, I think I just maybe at this point just purely luck. I think I've been extremely fortunate to have a group of very talented young people. I think I don't hear or have to deal with any conflict. I don't think they exist in my lab. Part of the reason I think is because we're working on very interdisciplinary research. So there are synergies between folks' research, but there is essentially no competition within the lab. So everyone is just, focusing on their own thing and helping others instead of trying to sabotage each other's project. I know if a lab is more, if the research area is more narrowly defined, and sometimes the PI also does terrible things by assigning several people on the same project, that obviously can cause conflict. And I would urge everyone, if you do enter grad school, you do want to, I don't know, not gossiping, but you want to check with the grad students in the lab and see if they're happy, if they're assigned on project that someone else is also on. If that's the case, I would say, run away from that type of lab.
Time for about one more question. Someone has it?
If not, okay.
Oh, wait.
Wait, so, okay, so you said you went from chemistry to material science, so was that mainly just because that was how you could get into that person's lab more, or what did you just prefer material science, like by content?
Well, the practical reason was material science was my PhD advisor's home department, so it was easier to place me in there. But I think once I joined that lab, I also realized, there's a reason that's his home department, because the research focus is definitely more materials oriented than chemistry. He is also affiliated with chemistry, so these things are connected, and his research is also interdisciplinary. But for me, I think it was initially for practical reason, it's easier. But later on, it also the research team also fits better with materials.
Dr. Guan, it sounded like you were about to say something before that.
I was
question.
gonna
Did you have a
say,
final-
if you don't have questions now, you can always reach out to me later. Yeah,
Thank you for that, and thank you for coming to visit us.
of course.
Sharing your wisdom.
Anytime.
Appreciate your time. I will stop the recording now.